Welcome to the CannMed Coffee Talk podcast, where we talk with the leading minds in cannabis science, medicine, cultivation, and safety testing who will be part of the CannMed 2020 conference in Pasadena, California this September 20th through 22nd. I am your host, Ben Amirault. I'm the marketing manager of Medicinal Genomics and a proud member of the CannMed 2020 team. Head over to CannMedEvents.com to check out our Phase 1 schedule, which includes presentation topics from 20 of our invited speakers and abstract presenters. We will continue to add to that lineup throughout the summer, so be sure to sign up for email alerts so you don't miss any announcements. If you sign up on our podcast page, CannMedEvents.com/Coffee Talk, you will also be entered into a drawing to win 2 tickets to our CannMed 2020 VIP dinner. On this episode, we talk with our CannMed 2020 Cultivation Keynote presenter, Seth Crawford of Oregon CBD. Seth and the team at Oregon CBD specialize in turning out new, exciting, and unique industrial hemp varieties designed for maximum yield and large-scale production. Their early adoption of disruptive testing technology and large-scale breeding techniques allowed Oregon CBD to produce a number of industry firsts, including the first high cannabinoid content auto-flowering CBD plant and the first pure CBG, CBC, CBDV, CBGV, and CBCV field-ready varieties. His presentation at CannMed 2018 was an instant hit, and as I mentioned in our conversation, Seth was mobbed with questions from attendees immediately following it. I know because I was trying to get an interview with him and had to wait for the crowd to clear. We dig into some of the details of that CannMed 2018 presentation, particularly the process his team uses to develop unique, stable hemp seed varieties. Seth shares his thoughts on where the hemp industry is headed, as well as some of the challenges the industry is facing this season. I also noticed when I listened back to the recording that Seth dropped several references to the year 2021, which could offer some hints into what the keynote presentation he has planned for CannMed 2020, which happens to be titled Cannabis 2021, The Golden Age of Discovery. We are excited to have Seth as a keynote at CannMed 2020 this year, and we are excited to see you there as well. Go to CannMedEvents.com now to see our ticket pricing options and get your ticket today. We are currently offering early bird pricing through August 1st. We are also offering a 100% refund to ticket holders should CannMed 2020 need to be postponed due to COVID-19. Rest assured that we are working closely with the Pasadena Convention Center to make sure that we can provide a safe environment for our staff and attendees. And as of this recording, the event is still on. But just in case, we have updated that refund policy. Go to our website for more details. That's CannMedEvents.com/COVID-19. I also wanted to thank this episode's sponsor, Advanced Nutrients. Founded in 1999, Advanced Nutrients was the first to develop a complete nutrient system that unlocks the true genetic potential of the cannabis plant. Since its inception, the brand has introduced more than 50 innovations to the cultivation community and continues to revolutionize the space through proprietary scientific discoveries. Their most recent innovation, Advanced Hemp, is the first all-in-one fertilizer that is backed by 20 years of science and specifically formulated to assist growers who are looking for higher yield CBD counts. For more information, visit advancednutrients.com. And finally, we're proud to say that this episode of the CannMed Coffee Talk podcast is fueled by the Hemp and Coffee Exchange. The Hemp and Coffee Exchange is a grassroots Tampa-based coffee roasting company creating progressively sustainable consumables combining 2 of the most genetically diverse plants on the planet, hemp and coffee. Hemp coffee is a healthy, delicious, natural product rich in trace minerals and nutrients, providing sustained energy without the crash of regular coffee. For more information, check out hempcoffeeexchange.com and use the promo code DRINKHEMP to get 10% off your purchase. Okay, without any further ado, please enjoy my conversation with Seth Crawford of Oregon CBD. Hey, Seth, thanks for joining us on the podcast.
Thanks for having me, Ben. I really appreciate it.
Of course. And let me say, the team's really excited to have you as our keynote presenter for the cultivation focus area for CannMed 2020. Your presentation at CannMed 2018 was a standout for sure. And I might have a question or two about that, as I'm a novice when it comes to plant breeding, but I'm still very interested in it. And if others are interested, and I imagine you are if you're listening, but I encourage you to listen to Seth's CannMed 2018 presentation. I'll put a link in our show description so you can check that out. But if I remember correctly, and you could tell me, but I think you were mobbed after the talk with people asking questions and maybe handing you business cards, which is always a good sign. So we knew we had to have you back as a keynote, and that we were sure that you'd have new data to present. So I'm wondering, we're dying to know, what is it you're looking to present at CannMed 2020 this year?
Most definitely mobbed. It was fantastic to get that kind of response from folks. I think in a lot of ways, what we're doing up here in Oregon, and there's other folks around the world that are engaged in this in similar levels as us. But what we're doing is really dramatically different from previous cannabis breeders, and the things that we've been lucky enough to uncover both on a phenotype and chemotype and now even genotype level is just distinct, right? It's different from what's available, and we're continuing to push that forward. So this year we're really excited to be able to talk about some of the breeding projects that we're working on. Obviously, both the collection of the phenotype data, chemotype data, genotype data. We're going to give you a little bit of an update on our collaboration with Oregon State University and how that's going. There's a number of very interesting fronts there. We're going to look at some larger population genomic studies that we're engaged in. Both remember when Kevin was on talking about his presentation, the Eurofin SNP chip. We're participating in that with some of our F2 varieties, along with some competing approaches that are really looking at identifying structural variants that are present in the cannabis genome, and specifically structural variants between individuals within the population, as that seems to be where a lot of the most uh, interesting examples pop up in terms of new genotypes that express new chemotypes and new phenotypes that are much more acclimated for specific types of production. So we're really excited to be able to share the findings. Obviously, we're still in the process of gathering all that information, but we think that it's going to be a significant revolutionary shift in the way that cannabis is produced based on a lot of the results that us and others are collaborating on finding.
Excellent. Now, are you still very much at this stage looking for, you said variations? Is it in regards to, cannabinoid expression, terpenes, and sort of potency sort of things? Or are you looking also at, actual plant structure or drought resistance and things like that?
All of the above, yeah. And again, any good breeding program, you really have to narrow in on things that are really critical for specific projects. So I would say that the answer to that depends on the project that we're working on. But we do have some awesome trials going on with OSU over the summer. We've got 12 different states as of this afternoon that are engaged in a multi-state land-grant university variety trial to be able to assess what kind of differences emerge in different ecosystems, whether it's from flower timing to disease resistance to water usage. Um, a number of other trials going on here in the Western United States, specifically focusing on water consumption. That stuff's all really interesting. But I think some of the biggest breakthroughs that we're going to be looking at for 2021 are really coming down to chemical data and chemical changes in the plants. As you're probably well aware, the USDA has made it fairly difficult for industrial hemp farmers to continue to grow large fields of cannabinoid rich plants, specifically CBD plants, in part due to the underlying genetic predisposition of the CBDA synthase to produce some residual THC. But we have actually figured out a way to basically get around that. And we've got some wonderful, wonderful plants that are very, very rich in cannabinoids, CBDA, CBDVA, CBCA, and have great olfactory characteristics as well. So these are plants that have wonderful terpenes and other compounds that are responsible for making the plant smell. I think this is going to be something that everybody comes to understand a little bit better in 2021. But yeah, we're really excited about these new chemotypes and being able to get them into farmers' hands to allow them to continue successfully producing low-cost cannabinoids for the public.
And now Oregon CBD is specifically focused on hemp, correct?
Yeah, I mean here I saw the in your list of questions that this was going to come up, and my PhD is in sociology, so I'm going to just step back and say any dichotomy, any sort of external definition that we place on the cannabis plant is a social construct first and foremost.
Ah,
There's more similarities between a plant that has a say a THCA synthase that's active and a plant that has a CBDA synthase that's active outside of those genes. Yes, those those two synthases end up determining what's legal uh, in most countries in the world. However, it doesn't necessarily demarcate an actual difference between plants. So everything's cannabis. We got into the industrial hemp space just because it didn't have the same plant count restrictions that the THC production space had, and we knew if we wanted to actually breed plants the way that you need to move this crop forward, you were going to have really really large numbers, access to an actual bank account,
mhm.
relationships with universities and other institutions, and that was really the way to move forward.
So when you say that the way that you're doing breeding is different than kind of traditional breeding that's going on in the cannabis space, is that what you mean? That you're able to do it at this large scale and kind of do it the proper way?
Yeah, and when we say proper way, not just scale. So obviously, with any type of information, the larger your sample size is, the more accurate your underlying data is. In our case, we were really bucking the trend, and this is that it's present in the THC breeding side. And I won't say that all THC breeders are doing this, but because it's illegal and it's essentially still an underground activity, even for the folks that are operating in state legal production programs, there's always the overhang of federal interference, which means that you can't run huge numbers, and you have to focus on some very specific characteristics. You also don't have the freedom to do 5 generations of inbreeding with a specific variety before you outcross it to create your field ready versions. So in the THC side of things, a lot of times you see people that are taking sour diesel and crossing it with purple kush, and the resulting plant is purple diesel or sour kush or whatever it happens to be. But in reality, the offspring of those two plants, well, you're going to end up with maybe 1 out of 20 phenotypes that's just amazing and great for production, and you can clone that and then grow your small canopy on the THC side. From a breeding perspective, you just created an absolute mess of genes. It's not going to produce a uniform crop. uh, we call it polyhybrid pollen chucking is the term that's used by people that are doing the actual line breeding. And line breeding is difficult. It takes years to do. The average development cycle for one of our varieties is about 3 years, and that includes, from the time that you actually identify your standout in a population to inbreeding it in isolation tents, self-pollinating repeatedly for generations, and testing each one of those generations to figure out which plants have the traits that you're looking for, both from a chemical, phenotypical, and now uh, genotypes as well, making sure that you're getting what you want. And then once you've identified 10 to 20 standouts, 4 or 5 generations into that process, you then outcross and perform field trials. And those field trials are where you actually get to see which of your inbred population, which of your inbred plants ends up producing the best field production populations. So we end up, in a situation like that, we may think we have 20 amazing plants, but in reality, we narrow it down to 1 or 2 that ends up becoming a production plant. Um, we throw away 99.9% of what we start with, and that's a different situation I think most of the folks in the THC world are accustomed to.
That's got to be heartbreaking in a way.
Sometimes progress requires death. It's the plant breeders on staff. We all, it's it's so funny. We all obviously deeply, deeply love plant life in particular, but at the same time, we know what our goals are. It's sad to cut plants down, but it's also to a certain extent a relief. You're taking the unknown out of the field for a farmer and replacing it with something that is going to be a stellar performer. So it's, yes, it's sad, but at the same time, there's a bit of joy knowing that you're actually making the gene pool better.
And are they just completely scrapped? Is there any way
Stacks. Oh man, stacks and stacks of plants. Stacks of plants. Yeah, no, they're gone. They're done. We obviously we store DNA to go back and figure out, hey, what was wrong with this particular phenotype, genotype, chemotype, etc. But no, we literally we compost them.
Okay. Now I want to step back a bit because you were kind of talking about your process of, taking creating a new variety, and you had a great slide about this in your presentation in 2018. But I'll fully admit I got a little lost, so I don't know if you can kind of help me. So, so you start with your 2 parent lines and you get your F1s. Take me through from there, because there are some terms like backcrossing and selfing that I'm just not familiar with, but I'd really like to try to kind of understand the process here.
Yeah, most definitely. And on that slide, what I was basically starting off with were 2 very diverse parent plants and what it would take to actually go through and identify segregation in your resulting population. The more common approach is to take 2 plants that you think are distantly related and inbreed each one of those lines repeatedly over and over and over until you get to the point where you have a highly homozygous plant. It's basically it's homozygous at most critical loci in a way that leads to predictable phenotypes and chemotypes.
So let me stop you there. So you have
Yeah,
the 2 plants, you cross them, you have a generation after that.
let's
So when
yeah
you're
step
saying that
step
go
back
ahead.
from that for just a second. Let's say you find a plant that has a great chemical profile, regardless of where it came from. Uh, the next step in that process is to self-pollinate that plant. So that
Got
plant's
it.
going to get inbred, and then you're going to pull the seeds off of that plant, and you're going to grow them out and do exactly the same thing. Identify the ones that have the characteristics that you're looking for, self-pollinate those, collect the seeds, and basically start the process all over. At
Okay, so
the
you're
end of,
continuing
say,
to self-pollinate. You're not actually crossing it with, or crossing it with a sibling or anything. It's all through self-pollination.
that's the quickest way to achieve homozygosity.
Okay.
So what you just described, say you've got a great chemical result in the initial plant that you have. Another way to do that is to take one of your self-pollinated seeds and then back cross it to that original mom that had the right chemical profile. We tend to stray away from that in part because of what I was describing before. The fact that most of the cannabis varieties that are publicly available are very, very heterozygous. So they're a total mess, and that self pollination ends up leading to less variation and a tighter likelihood, much higher likelihood that you're going to get what you are actually looking for. So to take that and then back cross, sometimes that works to lock in the trait that you're looking for, but more likely than not, with the current population of cannabis that's present in the world, you're going to end up with just a bigger mess.
Okay. So where'd we stop there? So we're in the F2s. Where do you go from
Yeah, that's where the magic happens. To be honest, that's in your S2 population or your F2. S2 is best because then you're not crossing siblings, and it's a strict self pollination regime.
Mhm.
You're going to find plants that are they have the maximum amount of diversity that's present in cannabis in the F2 population. You're going to see tall plants. You're going to see short plants. You're going to see plants with giant calyxes, small calyxes. you'll see a diversity of olfactory compounds that are giving them distinct smells.
So
And
just,
that
I don't mean to cut you off, but just to review. So this is the product of that original cross between the two distinct lines, and then that self-pollinated, that's creating the F2s that have a lot of different varieties. Do I have that right?
you can do it that way. Yeah.
Okay.
You can definitely do it that way. Uh, I guess what I was getting at is from a developmental standpoint, if you've got a plant that has the chemicals that you want, generally what you're describing right now is, give you a good example. We've been working on Varins, CBDV, THCV, CBCV, et cetera, for about 4 years now. And once we had finally found some plants that had very high concentrations of the varins, it turns out that they have really poor structure, not great smell, horrible flowering time. Like they're plants that you would just throw away normally. But it does have that specific ability to produce divernelic acid. So we take that plant and we cross it to a plant that has all of the other characteristics that we want, but is
Mhm.
missing the divernelic acid component. And in that F1, then we go through and we look for the plants that have the highest concentration, that have the best structure, et cetera. And then we
Mm,
go through exactly what you just described. Yeah.
got it. Okay, great. All right. So sorry, I didn't mean to cut you off or
No,
take your
no,
thunder.
it's kind of, I mean, this is the downside of audio. If we had some video on here, I'd throw some slides up for you. It would make a lot of sense. But
Right. All right. So you're in the F2s and you're seeing all this different variation. So then I imagine that you find the best performers and then repeat that process again?
exactly, yeah, you inbreed those, and basically we like to repeat as long as we possibly can. One of the issues that we run into is cannabis it's an obligate outcrosser. It really likes to outcross to other plants rather than to itself, and in a lot of cases these plants become sterile fairly quickly. And it that in and of itself is a inherited trait, and it's something that we select against at first in our populations. uh, but yeah, you basically just do it as much as you possibly can, with the goal of, I mean, we've heard this a lot lately, this notion of flattening the curve, quote unquote, taking a large, what would end up being a large distribution of people, and in our case traits, and spreading it out. So what we're trying to do with cannabis by inbreeding it is to reduce the amount of variability that's present. That's the curve that we're trying to flatten. You're trying to create something that doesn't have variation, so that when you do outcross it, you're going to have something that breeds true for very particular characteristics.
Great. So yeah, so explain to me outcrossing. So you're doing all this inbreeding to stabilize, and then you say that you do an outcrossing. What exactly does that mean?
That means you're actually making the F1 seed, so your true hybrid seed that will go into a field for farmers. That's like, it's the you've done all your homework and now you're doing the presentation. It's out in the world for everybody to see.
Got it. So you're basically just planting it in the field.
Yeah, and what's required to make that F1 are two independently developed, highly inbred lines.
Okay.
And like I was saying before,
Got you.
you
Gotcha.
may have, like, you may have 20 plants in one of those lines that all seem great, but when you cross them to make your F1s, maybe they don't perform as well as, and this is always the case. There's one or two plants that just outcross exactly the way that you want them to, and perform better than all of their siblings. So yeah, two independent lines to create your one line that actually goes out to farmers.
Excellent. All right. Well, that was very helpful. Thank you for that. So you guys are constantly doing this, coming up with new varieties. What are some of the new and interesting ones you've come up with, or that you're the new and interesting varieties that you're excited about? What are they?
The big ones for 2021 are all compliant. So the USDA gave us this insane, well, it was Congress, but USDA is enforcing it. This scientifically inaccurate zero point 0.3 total THC concentration limit. And there's a lot of questions in the industrial hemp farming sector. Does that mean 28 days before harvest? Does that mean 14 days before harvest? Does it mean the day of harvest? Does it mean final products, et cetera? It's been really difficult for folks who are on the traditional CBD side of breeding to accomplish. We're really, really excited. In addition to our pure CBG, these are type 4 plants that we've been working on and been in various stages of field trials and public release since late 2016, early 2017. In addition to those, we now have type 3 plants. These are high CBD plants that even on the day of harvest and even after you've trimmed them and they're in a store or for sale online, uh, they are compliant, 100% compliant with that zero point 0.3 total THC requirement. In addition to having a number of different unique compounds that I think are going to be really useful for people and enjoyable, I think, on the consumption side as well.
Excellent. And how has this season been for you? You mentioned the farm bill. Um, how has that shaped your season?
Hurry up and don't do anything is it's kind of been the mantra. We've been waiting for the FDA to issue some guidance. I know a lot of the manufacturers of CBD products have been waiting for some guidance from the FDA, and that has had a very depressive effect on the entire industry. Looks like there was some overproduction in 2019.
Mm.
We were basically being yelled at by farmers across the country for not being able to supply enough seeds for them. So we built out an infrastructure capable of producing enough seed to supply the entire country, and then the market shrank by about 75% this year.
Wow.
So we're fine. It has definitely been trying, and I know that farmers are in a similar circumstance as well, mostly because they're sitting on product from last year, and basically just waiting for the federal government to give the green light to have it out in the marketplace. Um, I think that's going to continue for a little while. We do know that Congress is willing to act to be able to tell the FDA, hey, it's time to move forward with cannabinoids, which I think is a very exciting moment. It's been a long time coming, but it's going to open the door up for other compounds as well. So not just CBD, but the whole pantheon of cannabinoids, including THC, to be able to get into uh, both the food supply and to be legitimate as something that people can consume.
Now, one of the things you mentioned in your presentation at CannMed 2018 that stuck out to me because I am a craft beer enthusiast was these cannabinoid-free varieties that could maybe be used in beer with the terpenes and everything as almost a substitute for hops. How is that going?
Really well, really well. They're really neat plants. In addition to being useful for beer flavoring, which to be perfectly honest, you could do with uh, other cannabis plants that have cannabinoids in them as well. But this just happens to get around a lot of the regulatory hoops. Uh, they're really cool. Um, they also are able to produce some other interesting compounds that go along with the terpenes and the other olfactory components that would be very, very useful for basically anti-inflammatories. So it's something that we're really excited about getting out, working on improving them for disease resistance and getting them out into the marketplace. The one thing that I would say though at this point is that the hop industry is fairly small compared to the cannabis industry. So it's definitely a niche market, but it's something that we're excited about just from both a craft brewing standpoint and then the union bringing hops and cannabis back together in some really important ways.
Yeah, and I know that you had mentioned that hop farming doesn't seem fun. What did you mean by that?
Oh, they're just 20-foot tall vines, and you have to replant 20% of all of your hops every year from downy mildew infections and that kind of problem. It's hard and very labor intensive to be able to get the components from a hop that are a lot easier to do with a cannabis plant, and you don't have the issues of long-term disease pressure popping up in your field. So it's less pesticides, less reinvestment every year for the farmer. And to be perfectly honest, cannabis is capable at this point of producing more of the compounds that people want from hops than the hops are themselves.
I know another thing that you guys have been working on is sort of autoflower varieties or early flowering lines. Sort of what's the difference between those two terms? And then I guess a follow up to that is, will photo period crops kind of be a thing of the past?
I think true photoperiod crops, I should hesitate on this. They have a place, but you have to understand the underlying genes that are responsible for flower timing and to be able to identify the variants that will get you where you want to go in specific places. So photoperiod plants will have a future, but it's more about gene stacking and being able to use those to get you into a period where harvest is going to be preferential. So like a place like Tennessee or Kentucky, for example, they have a very wet August and September, and then everything just kind of dries out in early October. Again, with climate change, this is obviously going to fluctuate a little bit, but for folks like that, you want to have the ability to harvest early and then also harvest late to maximize the usefulness of your space. Um, so I think there is always going to be a place for photoperiod plants. To step back, the difference between a autoflower and a photoperiod plant is one is considered day neutral. That's the autoflower. It'll flower under 24 hours of light. The photoperiod plant is flowering based on relative day length. So what we found, so we created the first in North America, the first autoflowering CBD plant in 2016, and created the first autoflower photoperiod hybrids in 2016 as well. Did a bunch of field trials in late 2016, and then again in 2017 to demonstrate the efficacy of combining these two distinct germplasm lines. And what we found was really interesting and driven home in 2018 even more so. When you take these hybrids containing an autoflower gene and a photoperiod gene, when you take these hybrids anywhere in the country, they all react to photoperiod changes in exactly the same way at exactly the same time. There's some slight variations depending on which photoperiod gene you have. But what it looks like is that these early flowering hybrids, you can clone them, you can maintain them, but they go into flower roughly 2 weeks after the peak of summer. So early July across the country, all of these plants go into flower. And what it looks like based on the literature is that this is basically creating a little bit more sensitivity to a shift in red light spectrum. So the plants pick up on that and go into flower. So some pretty good examples in the literature about mechanisms in other model organisms using these same genes. But we just happen to pick up on it and be able to use this in our hybridization process to be able to create these F1s that are predictable in flower timing across the country. And that's a huge advantage to farmers. What folks figured out after the first couple of years is that if you have a plant line that was developed in Colorado and flowers consistently and it's done by early October, if you take that to a different photoperiod regime like Florida or Vermont or Oregon, the plants respond in a very different way, and you'll end up, not finishing until December in Oregon. So being able to have that predictable flowering time is a huge benefit to farmers, and one of those great examples of where more knowledge that gets generated by everybody who's engaged in this process in the cannabis space ends up leading to much better products and better outcomes for farmers.
And again, this is particularly useful for outdoor cultivation, which, correct me if I'm wrong, is much more common in the hemp space than the cannabis space. And why is that?
Uh, Cost of production. It's free sunlight, free soil for the most part. The cost of production outside is much lower than in the indoor space. And to be perfectly honest, the only reason why indoor cannabis is still grown outside of producing seed for large scale farmers on the THC side, it's only because of prohibition. As soon as you drop that requirement and strange requirements like 12-foot tall fences or an enclosed space and those sorts of things, the total cost to produce outside just makes it much more likely that people will be engaging in that type of production behavior.
Now, so at Oregon CBD, you guys seem particularly focused on, resinous big buds producing cannabinoids and terpenoids for, whether it be supplements or pharmaceutical applications. Are you guys interested at all in maybe going after the other markets of, using hemp as a fabric, a plastic, a fuel, building material, anything like that?
Yeah, very much so. Although, the main take-home message on this is that other countries, because they had legalized industrial hemp ahead of the United States, I think they're a little further along in terms of their breeding programs for things like fiber or seed. I think that we have the tools to be able to catch up relatively quickly. And some of the things that we're really excited about for 2021 is being able to introduce new varieties that can do those things, particularly on the seed side, for consuming seed as a protein source, as a basically as a nutritional supplement in and of itself, as a food, and then also being able to grow your cannabinoid rich crop right next to that, and being able to safely do that without having any seeding.
Yeah, that was one thing I wanted to ask you about because if I remember correctly, you had an Instagram post sort of about being good neighbors and being careful not to grow male hemp outside. So I was wondering if you could kind of talk about some of the dangers that come with that.
Yeah, if your crop gets seeded, you're going to lose 30% of your overall cannabinoid content. You're going to lose 50% of the weight of your crop, just because the plant's putting all of its energy into seed production rather than cannabinoid or floral tissue production. Yeah, hugely problematic. It makes the production of certified seed outside also very difficult. There's a number of different issues that come along with it that farmers really, at the end of the day, they're just trying to grow a crop that's going to make them money and it's going to benefit people. And it's something extra that they shouldn't necessarily have to worry about. We are very confident that in 2021 we're going to be able to release seed lines that make that concern obsolete. Like we've figured out a way to get around that, and the result. We're so excited about this. The result is, I think, going to push forward the envelope of what people even consider possible with cannabis. It's the early results that we're seeing in our experiments are the most resinous, the most pleasant smelling, most vigorous plants that we've ever experienced, and we've been growing cannabis for pretty close to 20 years now. Just amazing evolutionary jumps forward, and we're really excited to be able to talk about this in depth at CannMed 2020.
Well, that's a great place to stop if I've ever heard one. But before I let you go, I want to give you a chance to plug your website or social media or anything. Where can people learn more about you and Oregon CBD?
Yeah, our website is OregonCBDseeds.com, and we are on Instagram and Facebook as Oregon CBD. We're 41 strong scientists, plant propagators, folks that are taking care of greenhouses. We've got a great company, family owned and operated, and we're just really excited to be providing farmers with quality seeds that they can depend on.
Excellent. Like I said, we're more than happy to have you, and we're happy to work with Oregon CBD. We're, as you know, a family operation over here as well, and I think we kind of see ourselves as sort of cut from the same cloth in the regard that we both try to do what we can with good science and good old-fashioned hard work. So again, happy to have you as part of CannMed 2020, and we'll see you out in Pasadena this fall.
Sounds great. Thanks for having me, Ben.
I hope you enjoyed my conversation with Seth Crawford. I always learn so much talking with him. Check out the links in the show description to learn more about the topics we discussed. And please also check out OregonCBDseeds.com. The Oregon CBD team has added some great content resources lately, and they're definitely worth a read. Next episode, we welcome Brianna Cassidy of CDX Analytics. Brianna is the lab director at CDX, a full-service cannabis testing lab in my hometown of Salem, Massachusetts. We discuss the challenges that come with testing cannabis flower, including sampling bias and the lack of standardized methods. Brianna also shares her approach for evaluating new testing technologies, a topic she will explore in more detail at CannMed 2020. That episode will drop June 24th, 2 weeks from this episode. In the meantime, please go to canmedevents.com/coffeetalk and sign up for email updates. That will enter you into a drawing to win 2 tickets to our CannMed 2020 VIP dinner and keep you up to date with all things CannMed 2020. If social media is your thing, you can find us on Facebook, Instagram, Twitter, and LinkedIn. Just search for CannMed Events. And lastly, if you are listening on a podcast app, go ahead and hit the subscribe button so that new episodes automatically download to your device. And if you want to leave us a 5-star review, that'd be great too. All right, that's it from us. Stay safe, stay healthy, and be sure to come back for the next episode of CannMed Coffee Talk.